Hello everyone, my name is David Sischo and I'm a wildlife biologist with the Hawaii Department of Land and Natural Resources. My co-author on this study is Dr. Michael Hadfield with the University of Hawaii. I'll be talking to you today about a species of land snail that has recently been described from the island of Rota. This species is new to science, but has been hiding in plain sight all along. This is work that I conducted at the University of Hawaii as a grad student. I'd like to mention, as we're starting out, that besides arthropods, which are insects, spiders, land crabs, etc., land snails make up a significant portion of the terrestrial biodiversity on islands. This is particularly true for Pacific Islands, where there are over 6,000 described species of terrestrial snails. Today I'll be focusing on snails in the family Partulidae. These are large, typically tree-dwelling species that occur throughout the tropical Pacific, from Guam and the Northern Mariana Islands in the Western Pacific to French Polynesia in the Eastern Pacific. Interestingly, they never occurred in the Hawaiian Islands. Unfortunately, Pacific Island land snails are also the most extinction-prone animals in the world. Habitat destruction, climate change play a role in this decline, but the most insidious and urgent threats come from introduced invasive species that directly prey on the snails. In the Mariana Islands and pretty much throughout the Pacific, it's the same suite of introduced predators. We have rats, which directly feed on larger snails. We have Euglandina rosea, which is an introduced species from Florida, which is a mollusk predator, and it was brought in as a biocontrol to control other introduced crop pests, like the giant African snail. And more recently, we've had another introduction across the Pacific, the New Guinea flatworm, Platydemus manowarki. And in some instances, and I think this is true in the Mariana Islands and Guam, this Platydemus has actually controlled Euglandina rosea and is now one of the most urgent threats for land snails. The Mariana Islands and Guam now have 7 species within the family Partulidae. This includes our new species, highlighted in red, Partula lutaensis, named after the Chamorro name for the island of Rota, which is Luta. Unfortunately, all but 4 species are thought to be extinct, and much of what remains has experienced range reductions and local extirpations, and now many populations are on the verge of extinction. Partula radiolata is a partulid tree snail endemic to the island of Guam. So endemic means that it occurs nowhere else in the world. Salmoana fragilis, the snail in the lower picture, occurs in very small numbers on both Guam and the island of Rota. And then there is Partula gibba, which has an unusually large distribution spanning 9 islands from Guam in the south of the archipelago to Pagan Island in the far north. This is an unusual distribution, as most species in the family Partulidae are single island endemic, so they have very narrow ranges that usually are single islands or even mountain ranges or valleys within islands, with a few exceptions that have multi-island distributions, but this is typically unusual for a Pacific Island land snail. And so this wide distribution was the inspiration for our study. We wanted to know if these multi-island populations were in fact all the same species, and if so, what is the distribution of genetic diversity? So over the course of our study, we were lucky and were able to jump on various surveys and collect 172 genetic samples, non-lethally collected genetic samples, I should say, where we took a very small sliver of the snail's foot as it was crawling across a clean petri dish. And so these samples spanned 3 species and 6 islands from Guam all the way up to Pagan. Back at the laboratory, we extracted DNA from our tissue samples and sequenced 3 genes, and these include cytochrome C oxidase subunit 1, CO1, and 16S from the mitochondria, and 1 nuclear gene known as ITS2. Then we used standard phylogenetic analyses to reconstruct the phylogeny or family tree, so to speak, of the Mariana Island partulids. We ended up with this phylogeny, and if you take a look at it, I'll give you a minute. You can see down at the bottom there's a cluster of Partulagibba from most islands, so this makes a lot of sense. All the different populations of Partulagibba are clustering together. But then if you move up to the second set of branches, you'll notice that all of the Partulagibba from Rota came out as a sister taxa, or a sister branch, to Partula radiolata from Guam. And so this was curious. We first thought we had made a mistake, and we resequenced samples to just confirm that this was actually correct, and it turned out to be correct. And so this was our first inkling that we might have a different species on our hands. And I'll just show you here. There's the Partulagibba from Rota. And if they were actually Partulagibba, we would expect them to fall out as in one of these branches down here with the Partulagibba cluster. To make things more confusing, Partula lutaensis had been formally referred to as Partulagibba, and also was kind of unofficially called Partula langfordi, which was an endemic species to Agiguan that is thought to be extinct. And so we kind of had to reconcile all of these different names and kind of prove that Partula lutaensis was in fact not gibba and not Partula langfordi. Partula langfordi is a small Partula species that was described as an endemic species to the island of Agiguan. In the 1970s, this species was described based on shell color and shell length and width, which differed from Partulagibba, which also existed in mixed populations on the island. There was a lack of intermediate forms between Partulagibba and Partula langfordi, with Partula langfordi being small and darker in color, and Partulagibba being larger and lighter in color. And so this was justification for describing it as a distinct species, even though the shell shape was largely the same as Partulagibba. It was just the size that was different. And so we decided, without having any usable DNA from Partula langfordi, to redo that analysis with the snails from Rota Island to see if there were overlapping shell sizes. And what we found was that, very similar to the Langfordi and Partulagibba on Agiguan, the Langfordi shell size, length, and width was similarly much smaller than Partula lutaensis on Rota, giving us some morphological justification for it not being Partula langfordi and actually being a different species. To further support our genetic and shell morphometric analyses, we looked at the internal anatomy of these Partula species using preserved museum specimens. And I promise that it is not just that all malacologists are a bunch of perverts, but it turns out that the anatomy of the reproductive system is a useful trait for distinguishing species. This is especially true when shell shape and size overlap, as in our case here. There were two traits unique to Partula lutaensis. This includes a vas deferens that is oriented downward compared to other species, and a larger expanded cecum, which is the chamber on the upper part of the reproductive tract labeled C, and this is also where the retractor muscle labeled RM attaches. So this internal morphology held true across specimens and offers more morphological evidence to back up our genetic evidence that Partula lutaensis is in fact distinct from other species. And so without further ado, I introduce you to Partula lutaensis. The shell diagram on the right is the type specimen for this new species that now resides at the Bishop Museum in Honolulu. And on the right is a photograph of a live specimen. These snails are exceptionally beautiful, existing in populations with mixed shell colors ranging from almost white to golden yellow, reddish brown, even banded like the live snail in the photograph. They're really beautiful animals. I'd like to leave you all today with some conservation implications and recommendations. Because Partula lutensis is an island endemic species, it is inherently at an increased risk of extinction just because it occurs over a very small geographical area. Additionally, we observed flatworms feeding on Partula lutensis as we were collecting genetic samples, indicating that they're likely under pretty extreme predation pressure. And lastly, looks can be deceiving. We must use genetics in addition to morphological techniques to distinguish species. So as new populations are found, we recommend getting a genetic barcode just to be sure that we're understanding what we're working with here. It's likely that other populations might pop up that aren't what we think they are simply because they have overlapping shell features that make them difficult to distinguish. And now I'll distract you with a petri dish full of multicolored Partula lutensis, and I'll take any questions you have from there. Thanks so much for listening.